The question is, what's happening with attendance? It's being collected. As you know, we haven't processed it, but it looks like it's working. We've got the first 16 or something, and it looks like it's fair and working. I think you guys even could review the first 16, and since then we've been doing the secret word, and that will kind of guarantee it works. So that should go. Okay, I have life. Maggie, you have your computer? No, email all the GSIs and ask why the hell they're not here. And yay. Maggie, as they straggle in here, can you just get them an exam? The microphone's not on, sorry. They both have a weak, they both have a dissociation constant at Ka associated with them. So we also, though, know something about bond enthalpies. And why do we know something about bond enthalpy? So I haven't given you in this section, in this particular question, have not given you an actual number, HBr, 150 kilojoules per mole for the bond, but you can still rank them, can't you? Because you can rank them by their polarity. The more polar they are, the stronger they are. So, and this is an easy rank in polarity, right? Just electronegativity difference. That's a higher electronegativity difference than this one. So you can rank those bond enthalpies, and could be given this, but I think we'll arrive at the same conclusion as well. This pKa of HF is a bigger, numerically bigger. That means, remember, the smaller the pKa, the bigger the dissociation constant is. So stronger acid for the smaller pKa. So this is the reaction that happens in solution for both of them. This is important to know. You should be able to write this without, I shouldn't have to write this for you. You should be able to write HA, weak acid, reacting with water to form H3O plus and A minus. If you can't do that, you will have a hard time on the exam, okay? I don't have to tell you this. You should understand how weak acids react with water. So let's just look at all the things we know from here. Pretty much everything from Chem 1 almost is on here. So we look at the relative bond enthalpies, and what we've been given here, we were given the plot for HBr dissociating. So this reaction, if it's HBr. And look at that, it is less than 0. So that is a negative number. So this HA, this dissociation constant, or this dissociation enthalpy, that is negative. This is a exothermic process. So we should have been able to take that home. And we should have been able to read that off the plot. Let me put that up there. It's not up there yet to remind you, but we can put it up here. The standard state free energy difference is the standard state enthalpy difference minus T delta S. Okay? So this plot, this intercept is the enthalpy, standard state enthalpy difference. So we can read off the plot that that's exothermic. We can also read off the plot that that is a negative slope. So how do you get a negative slope? Well, delta S better be negative, negative slope. Delta S better be positive. T's are always positive. So for minus T delta S to be negative, delta S has to be positive. So you can read those two things off the plot for this one. And we want to plot the other one, HF. So we have that from the plot. We have a why am I going K greater than 1 already? Okay. This is really good, isn't it? It's going to turn out to be a strong acid. Delta G, standard state delta G is negative everywhere. Standard state free energy is negative, K is bigger than 1. Okay? So all this is in here, kind of in intuition and understanding the relationship between K and delta G. Now I can actually classify that as a strong acid. That's the definition of strong acid. K, dissociation constant's bigger than 1. Because delta G is 0, that's a strong acid. For this one, how do I get that enthalpy is actually endothermic? Where do I get that? Well, I can start to look over here and say, I know these relative bond strengths, and I see I'm breaking an HF and I'm making an OH. So bonds broken minus bonds formed there. If this is a stronger bond, it takes more energy to break that than you get back when you make that. So you assume that's endothermic. Okay? Yikes, that's a lot in there, but important, but that's how I would arrive here. You assume their enthalpies are about the same. Or excuse me, entropies are about the same. It's just whether that's an F or a bromine or a fluorine. So that should be about the same. So you have everything you need to do it. You've gone through the whole analysis, and the difference is the slope's about the same, but for this one, you're going to assume delta H is a positive number, but same slope, because delta S will be the same. Okay? That is all that is on the exam. So that's good. That delta G changes with temperature, the sign of delta G. So at low temperatures, it's a weak acid. The acid strength gets stronger as you increase the temperature. Okay, but in this region, K is less than 1, it's a weak acid because delta G is greater than 0. Wow, that's a lot to digest. You might want to just digest that one after you're done here today. Go through that again, because that is all of the last half, really, of, or the last section of the class. And a lot of this beautiful thermodynamics, you know, this is my favorite thing. So that will be featured on the final exam. That's cool. Polyprotic acids, we've already gone through this. And we went through that. We'll just quickly go through this one more time, just to remind you, this is a buffer question. You have a pH of 7, but you know the pKa's. So if this pKa is 2, you're well to the basic side of this pKa. Very much more basic than that pKa. That means this will be the basic form. That proton will be gone. You're on the acidic side. You're lower pH than this pKa. So that means this proton will be on. Okay? The acid form dominates. Here, the base form dominates. Okay? So we understand that. pH is, the pKa is less than the pH. You're on the acidic side is how I remember it. If you're lower, you're more acidic. So the acidic form dominates. Here, you're on the basic side, you're higher pH, so the base form dominates. That proton will be off. Okay? So that's great. That's a fantastic review of acid-base chemistry. Here's ATP acting as an acid. This is adenosine triphosphate. This is the adenine, ribose, and the phosphates of adenosine triphosphate. One of the most important molecules in your body, as you know. This is where you, the body stores and transfers energy around the cell. So glucose is metabolized. The metabolic energy, the metabolic free energy, is stored in adenosine triphosphate and moved about the out of the mitochondria and into wherever it needs to be. But it's a polyprotic acid. The pKa's are around 6 for these and around 2 for that. So you can do the same thing at pH 7. They're mostly all off. That's on the basic side of all those pKa's. pKa 6 and pKa 2. So in solution, there's this highly charged molecule in your body, okay? Concentrated, compact, high charge. I just thought of a good question that won't be on the exam because it's already done, but this would be a fun one to add. I'll talk about it in a second. This bond here is what we saw a couple weeks ago, and they called it a high energy phosphate bond, saying when you break that bond, energy is released. We know that's not the case. It is the case that this hydrolysis reaction is exothermic, but if you take nothing else home from Chem 1, take home the fact breaking bonds always requires energy. Okay? So if I'm going to break that bond, I do it in conjunction. It's a hydrolysis. I do it in conjunction with a reaction with water, and you make adenosine diphosphate. This phosphate comes off. You get the free phosphate and proton. You change the pH when this happens. Free energy for that happening is minus 30 kilojoules per mole at body temperature, okay? So that reaction, it does release free energy, but it's because it's combined, it's a more complicated reaction than the just breaking of that bond. Other things happen. The enthalpy is minus 20. The entropy is plus 34. So both of these things favor a spontaneous reaction, okay? So what's true of spontaneous reactions? The entropy of the universe must increase. That's the definition. That coincides with free energy, standard state free energy is negative, is less than 0, okay? Those two things go together. One more thing goes along with it, because the free energy, standard state free energy is less than 0, K is bigger than 1. Products are favored. So all those three things should go together. Entropy of the universe increases, free energy downhill, products favored, K bigger than 1, okay? Take all those things into the exam with you tonight. Every single one of those is a question on the exam. But be careful. The entropy of the universe has to increase. The entropy of the system does not, right? It's not system entropy. So we know spontaneous reactions can go with a decrease in entropy as long as somewhere else in the universe the entropy goes up. You have to track the whole universe entropy. And you may not remember, but that was the beauty of the free energy change. That tracked the entire universe entropy for us. But system entropy could go up or down. Enthalpy could go up or down, and it could still be spontaneous. You have to look at the combinations of the enthalpy and the entropy. But remember, don't be confused. Just because, in fact, someone came into me one day and said, Mark, I don't get this whole entropy of the universe is always increasing thing. Entropy is not conserved. You know, take that home. It's not a conserved property. Free energy is not conserved. Free energy is the entropy change of the universe. It keeps getting bigger. We keep wasting free energy. someone came in and they said, Mark, I don't get this whole entropy of the universe is always increasing. Look at me. I am growing, strapping youth, getting bigger and more complex and more organized every day. How can that possibly happen in the universe? And I said, well, you are just the system. Yes, you are getting more complex and there's organization happening. But what are you doing right now? You're spewing a lot of hot air at me. You are taking in those metabolites, metabolizing, and then just blasting gases out into the universe as you talk. The entropy of the universe is increasing because of your metabolic processes. So overall, you can get more complex because the overall universe entropy is increasing with all the catabolic processes. The breakdown processes outweigh the metabolic processes of building up your cells. So all those things are on the exam in some form or another. Here is the hydrolysis in a little more detail. So there's the water molecule coming in. You have to make that bond and break that bond and make that bond, and the makings release more energy than the breakings, and you get a, so that's not the high energy bond, that is how you should say it to your biology friends and colleagues, okay? So let's do it in the form of a chem quiz, see if you've learned anything right there. Certain bacteria can live at 100 degrees C down in the ocean vents. Have you seen this? Tremendous pressure down at the bottom of the ocean. The temperature's above 100 degrees C because there's geothermal activity. It's heating the water. And it can superheat the water because it's under that high pressure. So the temperature of the liquid water is above 100 degrees. But they found bacteria living there, thermophilic bacteria thriving in tremendous pressure and tremendous temperature, where you'd think in the lab, what do you do? Oh, you might not know this, but in the lab, when you sterilize something, what do? You put it in what we call an autoclave. And what does that do? Pressurize and steam. You go to high pressure and high heat, and you try to kill stuff. These guys would just say, bring that on. Would thrive there. These would be dangerous bacteria to have loose in the lab. So, but can they use ATP? Can they use ATP as their power storage and transfer molecule? Well, let's look at it. I gave you these things. This is just the standard state number at room temperature. These two numbers, though, tell you what? These two numbers tell you that the plot, delta H negative, delta S positive, so the slope is negative. What happens? Is this reaction spontaneous at high temperature? Yeah, they can get free energy. In fact, they'll get a little more free energy for every ATP they hydrolyze at high temperature. So there was an evolutionary benefit to going to high temperature. You get more energy from your ATP hydrolysis. So there you go. Apply real world to your biology. Okay, here is that reaction. A couple things to think about this reaction for the exam tonight. One, energy is conserved. Energy is a state function. Take that home. If you take nothing else home from Chem 1, the energy of the universe at day 1 is the same as it is today. The total energy has not changed. All we've done is move it around and waste a bunch of it. We've turned perfectly organized things of high energy into disorganized things, high entropy things of low energy. That's all the universe does. This reaction here, energy is conserved. That means it's independent of path So remember, delta E is final minus initial, and that's it. So if this reaction happens in a calorimeter on the desktop, or it happens in your body, the free energy release is this, let me rephrase that. The energy, delta E, is the same. The same enthalpy is released whether you do it in, you burn sugar or a marshmallow in a flame, or you do it, your cell is doing it in the mitochondria. Same difference, delta H for those, okay? So let's look at it. It's also an oxidation reduction reaction. So we get to look at our redox chemistry. Here are the things that are happening. Here's glucose. It's a ring. So I've just drawn, didn't want to draw it as a ring. I just drawn this big line to indicate that carbon is attached to that carbon. But you can calculate, you know how to do oxidation numbers. Draw the Lewis structure. It's the ionic interaction. Oxygens are more electronegative. They take all of the electrons away. They leave carbon with no electrons. That gives it an oxidation number of +4. This oxygen, well, oxygen and oxygens, they share everything equally. That oxidation number is 0. But over here in water, oxygen grabs all of these electrons away from the hydrogen, and it goes to oxidation number -2. That you should be able to do relatively quickly from a Lewis structure, okay? So you see, carbon is oxidized. Its oxidation number went up. Oxygen is reduced. Oxygen is an oxidizer. You can see the definition of oxidation is also kind of clear. It's adding oxygens to things often is an oxidation. Here you have a carbon with just one oxygen. Here you have it bonded with 4 bonds to an oxygen. So oxidizing, adding oxygen. Reducing often correlates with adding hydrogens, not as often, but that's a reduction in oxidation number. Easy to remember that. So this is a redox reaction that's happening in your mitochondria. When you burn it in a flame or in a calorimeter, you just burn it and say, okay, there's this combustion, it's releasing energy, and carbon dioxide and water are being released into the atmosphere. How does your body do it? You may have this image that your body is a little steam engine, and there's a little fire in there burning up that glucose and sending it, you're breathing out that carbon dioxide and water, and somehow it's turning little turbines in your cells and mitochondria and making energy. Let's look at how it actually works. Here is a picture of a mitochondria, and it's just, this is a membrane inside the mitochondria, and the whole thing is a mitochondria, and there's a membrane inside the mitochondria. There's an inside and outside of that membrane. So there's intermediate space, inner mitochondria, and then there's this membrane, and then there's inner space. So this membrane essentially separates, segments your mitochondria. How does this happen? Well, inside that membrane, there are these giant proteins. And they're giant enzymes, and they're embedded in the membrane. How does it actually happen? So, inside the inner part of this membrane, sugar, there's your sugar, is oxidized down to carbon dioxide, but it's done in a very careful, complete oxidation without the presence of oxygen molecules. Okay? So it's an anaerobic oxidation. You formally oxidize, rearrange all these things. It takes 24 electrons to do that, and you can see you lose those 12 hydrogens when you do that. Okay? If you're just blasting H pluses into the membrane, into this space, what's happening to the pH of that space? It's getting more acidic. So the pH is decreasing. So it's getting more acidic inside than outside because this process is happening. Those electrons and a proton are transferred up to proteins in the membrane by things called cofactors. And you pump protons out using the electric potential of moving these electrons around. And you saw that. You can do electrochemistry. You can move electrons, and you can steal some of the free energy and do this work of pumping a proton across the membrane. At the same time, you have oxygen outside the membrane. This is what you thought would happen. These two things would encounter each other in your body somewhere. They're not. The oxygen on the outside of the membrane is coming through the membrane and reacting with protons inside the membrane to make water. So that reduction happens here. You can see it's a reduction. I'm adding these electrons that it takes to make that happen, to reduce the oxygen. So oxidation happening here, and you funnel the electrons over into the reduction. So it's exactly like a battery. You have the oxidation happening in one half of the cell, membrane, reduction happening in another part, and a salt bridge between them. So indeed, you are not a little steam engine. You are a little battery. Your mitochondria are little batteries working to power your cell. Is anybody a fan of the movie The Matrix? In The Matrix, if you don't know, has anybody not seen The Matrix? Really? Google that. It's like one of the most influential sci-fi movies of all time. In The Matrix, if you don't know, the end times happen. AI takes over, and we're very close, very close to this happening. So watch the movie now. The AI takes over, and they scorch the skies and to try and essentially kill all the humans. But the AI realizes I need energy, and if I torch the sky and I can't see anything, then I need to get power from somewhere. But the AI knows, oh, people are little batteries. So what they do is they take all the humans and they connect them to a giant in series and take the electrical energy out of them, and they use that to run their computers because that's how they have to survive. They need the electricity from people. They find out, though, that the people in their little battery pods, the human beings just die if they don't provide their mind some stimulation. So they provide this artificial reality. And the artificial reality is what the human beings experience. And one of the points of the movie is that could be happening. This could all be a simulation, and we all could be in a battery farm somewhere powering the AI that's powering this simulation. So, spooky. But here's the thing that you can take home and tell your friends while you're watching the movie. The way the AI keeps the people alive is when a body wears out, they just digest it down to soup, and then they feed it back to the other bodies. But that won't work because free energy is not conserved. You need the free energy, and if you use free energy and you build up a body and then break it down and try and feed it to another body, you lose the free energy. It doesn't matter. Entropy will continue to increase, and the matrix doesn't realize this, but they will run out of energy eventually. You can't just feed the same compounds back into the system. Okay? So you are batteries, but there's popular culture and there's thermodynamics involved there. So this is a beautiful thing. Is there any other animation here? Oh, this is very important. So what's happening here is you're pumping out protons, and then you are using up protons here. So you're keeping an overall H+ deficit here by pumping them out and then using them up for the reduction. So the pH is actually increasing. You're using up all these H+. So now you let them, at a different point of the membrane, you let them pour back in. And they go through a different giant enzyme. And this giant enzyme attaches a phosphate to an adenosine diphosphate and makes ATP. And that is the capture of the free energy of this oxidation reduction reaction. Okay? So your body is a little battery that's powering a proton pump, and the proton pump makes ATP from ADP. It's actually, can we look at that? It's worth, oops, something's happening. Oh my goodness, what kinds of things are happening? You can actually, here's an animation of that happening, hopefully. Got enough, there's that giant enzyme. This is the membrane.
Which causes the rotor-like motor to rotate.
You're gonna have a hydrogen ion on the outside being pumped through.
Then exits the intermediate space through the A subunit.
It's pumped out like this, and as it pumps out, this whole complex enzyme churns. And
which causes one of the beta subunits to loosely bind.
Here comes an ADP and a phosphate, and this giant thing actually turns on the surface of your mitochondria and connects as it turns. As that proton pump is running it, it uses that mechanical energy, turns it into chemical energy to make your ATP.
And axle to rotate another 120 degrees, which promotes the formation and release of ATP, after which a new ADP and free phosphate
Amazing.
bind to that unoccupied beta subunit. This
So
process continues to produce 1 ATP for every 3 hydrogen ions transported from the intermediate space into the matrix. Also,
into the matrix, it even said it.
Occurs in each of the 3 beta subunits.
That is freaking amazing. The reason they know that's happening is there's actually electron microscopy of that.
The next lesson in the Dirty Medicine Biochemistry series. In today's video, we're going to talk about oxidative phosphorylation.
Hush.
This is probably
So if you Google that, you will actually see electron microscopy of that little engine, that little proton pump moving on the surface of a mitochondria. Absolutely spectacular research to determine how that whole thing worked. We understand it in terms of almost everything we've talked about. Remember I said the first day, we have to use everything we'll learn in Chem 1 to understand how your body uses glucose. And here it is. It's the oxidation chemistry. It's the idea that oxidation chemistry can be coupled to batteries. It's the idea that pH is mediated by the number of protons. You can have a pH gradient, and then you have this entropic force. There's too many protons out here. You're pumping them out there, and you're using them up in here. And then entropy says, I want to equalize those concentrations. So I have this entropic force that forces the protons back through this proton pump and makes free energy out of an entropic process. So electronic energy, redox, batteries coupled to an entropic force are storing free energy in an ATP molecule. Absolutely gorgeous. It makes you want to go into MCB almost and learn about molecular and cell biology. So there's a picture, an animation, electron micrograph that's been colorized that shows those two regions. So there's the outside space, and then inside the blue membranes is the inside space that we were talking about, and all that membrane is littered with these protein complexes that are doing that chemistry. And we understand this, though, now. We understand how the pH will differ between the space, the red space, and the blue space, essentially, across the membrane. We have here, up here, there's excess H pluses up here. They're excess because we're pumping them out, and we're using up H pluses in here to reduce oxygen to water. So we get excess H pluses up here. If you have more H pluses up here than in here, more H pluses means lower pH out here than inside. Okay? So that's a question that you could do and understand about biology now that you're there. So we look a little bit more of the thermodynamics. So you can take that glucose, releases about 3,000 kilojoules per mole when you do this process. We know this process, though, now happens in a very complicated and beautiful way, but it's coupled to making ATP out of ADP. So you break down stuff, you couple that free energy. It takes, from a mole of glucose, you get about 38 moles of ATP. So you make 38 of those ADP, ATP, those phosphate bonds that you're going to hydrolyze later, and you're going to release that energy, and you're going to take, and you're going to grow. You're going to make free amino acids into proteins. You're going to make cell walls. You're going to do all the things that your body does. Okay? These two things, of course, those energy changes are the same. Energy is a state function. It happens in the lab desk or in your body, same release of energy. So you get about 4 peptide bonds, takes a ridiculous amount of ATP. So you know peptides, if you're going to make a protein, there's thousands of peptide bonds, thousands and thousands. And it takes 38 moles of ATP to make 4 of them. So millions and millions of ATP being hydrolyzed to make just one protein in your body. And of course, there's billions of them. Okay, so here we make it obvious. We have 2 things happening. We separate it into half reactions, just like a battery. In this cell, H2O is going, O2 is going to H2O. That's a reduction. Here, carbon is being oxidized. But all that energy, all the carbon dioxide could be burned in a calorimeter, and you know the answer to this one. It doesn't matter the pathway. This is one of the most, this is probably one of the more complex pathways you could possibly imagine for doing that reaction. And this is the simplest one. Put it in there and light it with a match. But the energy release is the same. Energy is a state function. I can go this crazy complicated path or this straightforward path, and there is no difference in energy, total energy released. Okay? Important take-home lesson. That's amazing, amazing thing happening in your body right now. Okay, that's all the stuff I had to review. I can take some questions. I have some other chem quizzes set up here that go over other fundamental things. But are there any burning questions that you need to know before you go in to the exam tonight? On anything from the entire semester? Anything at all? Don't do relationship advice, but other things, yes? Shielding. Good question. Yeah, there's some good shielding questions on the exam. When we talk about shielding, the idea here, the take-home lessons for shielding are, if you have a nucleus and it's got some plus charges on it because it's a nucleus, if you take nothing else home from Chem 1, the number of protons in the nucleus tell you what? The atomic number. That means the identity of the element. No other thing gives you the identity of the element. Not the mass. A lot of mass numbers up there. The identity of the element is only the protons in the nucleus, not the neutrons in the nucleus, not the electronic configuration. Every atom essentially could theoretically have the same electronic configuration. Our picture of atoms is they all look like a hydrogen atom, orbitals, just more electrons. Okay? What changes is the effective charge on the electrons, and that's where the this comes in. So you have, around that nucleus, you have orbitals. So you have s orbitals, and that's all this. An s orbital is, I'm not even going to draw the picture because I don't want to even ever draw it on the board. I'll just say it though. Electrons do not orbit a nucleus. We take that big take-home lesson. It doesn't happen. In fact, I'm not drawing it as a picture. In the old days, and there are not many textbooks that do this anymore, or not many people, hopefully, that do it anymore, but we used to, when I learned it, first they would teach you about electrons orbiting, and then they would say that's wrong and say they're in wave functions. Educational tip, don't teach wrong things ever. That's not a good thing. Don't spend a week of someone's time teaching them a wrong thing. So I don't even draw that picture or even say that to you. Electrons are a standing wave. They're a mishmash of matter that cannot be pinned down just doing this crazy distribution of matter around the atom. There's no nodes in that s orbital. So there's a probability, actually a high probability, of finding the electron right at the nucleus. There's no node there. The electron could be found right there. And that has tremendous impact. S orbitals, if you have an s orbital and your electronic configuration is 1s2, 2 electrons in that s orbital, those electrons have access to the nucleus even if there are electrons in p orbitals as well. Okay, and I've drawn 2 colors there. Why is that? Because there's a plus and a minus amplitude for a p. There is a node at the nucleus for the p electrons. The p electrons can't get in there. So it turns out that s electrons can hide, they can shield some of that nuclear charge from the outer electrons. So take-home lesson, s orbitals are very good shielders. And if you had to predict if this was your electronic configuration, and you're a neutral atom, if this is your electronic configuration and you're a neutral atom, that must mean you have 3 protons because you have 3 electrons. So you are lithium. That's what I've drawn here, 3 pluses. So I have 1 electron out here in the p, very well shielded by the electrons in the 1s. That means this p electron sees closer to just +1 nuclear charge than +3, because these 2s electrons shield 2 of those pluses, and the p electron just effectively sees just 1 positive charge. That's very, very hand-wavy. We say that the effective nuclear charge helps determine the ionization energy. The ionization energy is the effective nuclear charge. So this effective nuclear charge determines the ionization energy. But I never ask you to calculate it, okay? And I never give it to you and ask you to plug it into this. I ask you to rank it relative to other things. So if you had this configuration, let's do this. Let's do the excited state lithium. So lithium in 1s1, and we're going to put 3p2. So I excited it a lot. I put some photons in, and I went from n = 1, where that electron was. I bypassed 2, and I put it all the way up here in 3. So now I have, of the 3 electrons that I had, 1s2, 2p1, I took an s and a p, and I put them up there. Excited state. Still 3 electrons total, but this is the excited state lithium. I put in photons, made that excited state. Okay? If you look at the effective nuclear charge on these, you would say, well, it's probably close to about +2, because there's 1 s electron effectively shielding those p electrons. If you let that relax back down, so now you just have 1 electron up there in the 3p. This ionization energy is very similar to hydrogen in the 3p1. It would have empty 1s. This is 1 nuclear charge. That's hydrogen. It only has +1. This has 3 positives, but they're being shielded by these 2 negatives and give you about the same ionization energy for those. Okay? So that's kind of how we ask it on the exam. Look at things, how many s electrons are shielding p electrons, and you can say, well, these have about that p and that p have, this has exactly 1, and this has effective about 1 nuclear charge. So this is about 1 for both of them. They're both in level 3. So they have both the same ionization energy. Okay? Beautiful. That is all on the exam. Every single thing I said. Okay? Anything else? Other burning, interesting topics? You seem either exhausted or prepared or shy. Let's see. Anything else I can think of saying? Maggie, should I add anything? You just read the exam, didn't you? Anything? What bothered you most about it? Maggie thinks it's very fair. And Maggie's a hard ass, so if she thinks it's fair, it's probably fair. Maggie doesn't like to mess around. Ask me a question, I will answer it. Yes. Go over what? Acids and bases more? Yes. Well, geez, I think I told you every single thing that I want you to know already. Acids and bases, everything you need to know about acids and bases is contained in the titration curve. Okay? You should be able to write down, if I give you an acid like that one we just gave you, hydrofluoric acid, you should be able to write down hydrofluoric acid plus water goes to H3O plus F minus. That's a weak acid. That one's pretty weak. This one's pretty strong. HCl plus H2O goes to H3O plus Cl minus. Okay? So this is strong. K for this one, very much bigger than 1. K for this one, less than 1. Let's say we put those in 2 separate beakers. So here's a beaker with HF. Here's a beaker with HCl. And if I put the same number of moles of HCl and HF in there, which will have the higher pH? What? The higher pH. I know you always get that. High pH means basic. You got to reverse that in your mind all the time. Which one is more acidic? HCl, lower pH. Which one, the way you might ask it on the test is this kind of, I think it kind of appears on the test, something like this, now that I think of it. If I had to rank those 2 concentrations, what would I do? Concentration of H3O plus in that beaker and that beaker. Put the same amount of HF, same number of moles in each. What is that? Do I put greater than, equal to, or less than there? Which one? Less than. Yes, higher pH. Now I'm doing it. Lower pH in this one. Lower pH in this one, higher concentration here than here. This is a weak acid. It makes less. That's the whole point. It makes less of this. If both of these go, this one makes a high concentration, this one makes a low concentration. Okay? What if I mixed them up and I set them so they're both pH 3? Can I do that? Well, I'll tell you I can. I mean, you guys aren't chemists. But yeah, I can do that. I could add HF and HCl. But if I wanted to make this one pH 3, let's just do the calculation. Let's say I have a liter of water, and I'm going to put HCl in it, and I want to ensure that it's pH 3. What do I have to do? You have to, what makes it pH 3? H3O plus. If it's pH3, what is that number? Now I'm telling you what that number is, right? I'm telling you, what is it? Yes, if you can't do that, if you can't do this, pH3 means H3O plus is 10 to the minus 3. You've missed the fundamental point of this whole last week, okay? If I make it pH3, I could. That's what the H3O plus concentration is, 10 to the minus 3. So how many moles, here's a liter of this, how many moles of HCl do I have to put in there? What's that, Julia? Julia says, put 10 to the minus 3 moles in there. And then you'd have 10 to the minus 3 moles per liter. That's what that number means. This concentration means moles per liter. That's what that means. Put 10 to the minus 3 moles in there. And why do you know you need 10 to the minus 3 moles? What? Well, I told you it has to be pH3, but how do you know 10 to the minus 3 moles of HCl? You're right. 1 liter of water. You're missing the big point. It's a strong acid. Every mole of this you put in makes 1 mole of that. So it is 1 to 1. It's a strong acid. That's the whole point. Remember, strong acids, all their protons leave at once, and they're done. It's boring. Okay? This goes 100%. Every mole of that makes a mole of that. Put 0.3 or 10 to the minus 3rd moles of that in there. And that I can put on the exam. You know I would put that on the exam because you don't have to use a freaking calculator. You should be able to do all those steps. And I know this is summer. This sucks. You just were introduced to this 3 days ago, and now I want you to be sophisticated. But that's why I try to ask you the easiest things. That is the easiest possible question. Make HCl solution pH 3. That is the easiest thing I can ask. It doesn't seem that way in the first time you see it, but it is. Well, let me ask you another easy question. How much HF do I have to put in there to make it pH 3? How many moles of HF? And now, what? Now it's a weak acid. So now you don't get one of these. And I could ask you to do that, right? You could take the K and you could do, what do you guys call that, the icebox thing? You could do the equilibrium calculation and you could figure out how much of this I have to put in to get this to 10 to the minus 3. You could do that equilibrium concentration, right? You could say, I want, let's move some of this out of the way. If I want, H3O plus to be 10 to the minus 3, I want pH 3, then HF plus H2O goes to H3O plus F minus. I could say, well, I need this one to be 10 to the minus 3. Okay? If that one's 10 to the minus 3, what is, well, let's not go there. But I know the K for this, I would have to give you that, of course, Ka is HF products H3O plus F minus over HF. You know one of those, and you can lock together the other two and set it equal to K and figure out what you had to go to get there. Several steps, but I'm going to save them for you because I wouldn't ask you to do that. What I would ask you is the easier thing. I would ask you to say, is it bigger or smaller than the HCl I had to put in? Is it bigger or smaller? Do I have to put more HF or more HCl to get to pH 3? Way more HF. Okay, that's the fundamental thing to take home, because it does not totally dissociate. In fact, what are the concentrations at the end? Let's say it's pH 3. What are the concentrations at the end? H3O plus. They're both pH 3. Okay, the HF solution and the. So what's this concentration at the end? They're both pH 3. 10 to the minus 3. Okay, let's beat a dead horse there. What's this concentration? Also, you can read it off. What is it? Yes, they have to add up to 14. What's this concentration in the HCl beaker? Oh, conveniently. You put 10 to the minus third moles of HCl. You made 10 to the minus third moles of that, and it's the only place this can come from is the HCl. That's 10 to the minus 3. Okay? This is what you're doing. HCl makes that and that in equal amounts. Okay? Could have done that. Also simple. I try to ask you the simple things. That's one beaker. What is the HCl concentration? It all dissociates. You don't have any of this hooked together in the beaker. It all dissociates into that and that. Okay? These are the giant take-home lessons here. This is a homework question. You haven't gotten to it yet, but it's there. Can you do the same thing with the HF beaker? What's the HF concentrate? Well, okay, now we're going to do the HF beaker. What's the H3O plus concentration in the HF beaker? Everybody should be shouting it out. It's pH 3. We made it pH 3. Okay? HF concentration. How about that? Much bigger than 10 to the minus 3. I can say that. What is the OH minus concentration? Yes, still 10 to the minus 11th. It's pH 3. If you take nothing else home from Chem 1, the product of that and that are always 10 to the minus 14. Okay? But regardless of what's happening in water. Okay? What's that concentration? Yeah, should we try to bracket it? Every mole of this that dissociated made a mole of this. So you put a bunch of it in there. You put only a fraction of it dissociated. But every mole of it dissociated made a mole of that. You have a lot of this left over, but every mole of the dissociated made a mole of that and a mole of that. Okay, you just kept putting this in until this got to 10 to the minus 3. You kept putting it in, but every mole you put in, you were trying to make these, but every time you made one of those, you made one of those. Can't get away from it. Okay? All right? That's the kind of analysis you should do, and then you should be able to go to a pH, a titration curve. I don't know what the, I I don't know what the pKa of HF is. It's pretty small. It's something like 10 to the minus 5 or 6, yeah. The pKa of this acid, this HF stuff, is let's just say it's 6. It's low. It's a weak acid. So I get 1 point on the titration curve for free. That one. Sketch out the titration curve. Where am I? Am I at pH 6? No, I'm at pH 3. This isn't a very nice picture. That's pH 6 because I know that's half equivalence. pH is equal to pKa. Am I at pH 6? No, I'm at pH 3. That means I'm down here somewhere. And what is favored down here on the acidic side of the pKa? The acid form or the base form? The acid form, yeah. Acid form, very much greater than 10 to the 3, about 10 to the 3. Okay, acid form dominating here. Okay, so everything hangs together. Is the system in equilibrium right there? Is it in equilibrium right here? Is it in equilibrium right here? Yes, every single dang point, you couldn't measure the pH unless it were at equilibrium, could you? If the H3O plus concentration were changing, then your pH meter would never stop moving. It has to be in equilibrium. That has to have stopped changing. They are switching back and forth. They're jumping on and off those Fs, but overall it's not changing. Okay? Brilliant. That's the level of detail I want you to get to. That's why these 2 homework questions are on the homework. I want you to be able to do this kind of, well, I can get pretty close. Strong acids, easy. Easy, easy, easy. Everything dissociates. Easy peasy. Weak acids, well, you can calculate the numbers, but you can also estimate the numbers. Okay? And you know I would rather just have you compare them than calculate them. Okay? If you take AP chemistry, all you would do is you do icebox and you'd get that number. And you would take home nothing. But if you understand this, you understand strong and weak acids and equilibria and what pH means and titration. You understand all of that if you can do this rather than icebox. I think I can safely say, do we have to do icebox on the exam, Maggie? You just read it. No, there's no case where they had to do an actual equilibrium. There's no case where you had to even do an equilibrium calculation on the exam. Okay? I could make you do it, but I know you would be good at it. It's not hard. It's x minus x. Put them in the equilibrium expression. Solve for x. I know that you could do those steps. You could train yourself to do those steps, but you'd be wasting time. Not wasting time. I mean, you might want to know it someday, but I want you to learn something more than train yourself to do those steps. That's why we do it this way. Okay? Anything else? This was super, guys. You guys are going to be happy you were here. That's a lot of good stuff there. Okay, so you're happy you were here. I am happy that you were here. Semester's over. Good luck tonight. A couple things. All of you, I was excited to meet you 8 weeks ago, and I'm excited we got this time to get to know each other. After you recover, and in a year or so, you'll look back and say it wasn't that bad as well, and you will have learned something. All of you, please take this home, absorb what you can, go out and be those ambassadors of science. Some of you, I hope, will go into chemistry. All of you, move the world forward as ambassadors of science, as people who understand how things work. Change the world for and make it what you want it to be. Don't think to yourself, a couple people with a passionate idea can't change the world. That's the only thing that ever has changed the world. Okay? And you all can do it. So go out there and do it. Good luck tonight, and come see me sometime this fall. Take care, everybody.